Hi, this is Nigel Kooreman, here to discuss the outcomes of our study using induced pluripotent stem cells, better known as iPSCs, as a vaccine to target cancer. Our study has shown that our iPSC vaccine was very effective in priming the immune system to either completely fight off cancer cells or to significantly reduce tumor growth. Furthermore, as an adjuvant therapy after surgical resection of skin cancer, we found that our vaccine was effective in reactivating the immune system in fighting off remnant cancer cells within the resection areas. Overall, throughout the studies, we've paid special attention to the safety of our study, and we have found no adverse effects of the vaccine on the health of our research subjects. With the promising results of our iPSC vaccine to fight off cancer in animal models, we've taken the first step to test efficacy and safety on human samples in petri dishes, which will lay the first steps of moving this therapy forward to clinical trials. Just a little background. In 2006, a group of Shinya Yamanaka, Kyoto, Japan, first created iPS cells. They did this by putting embryonic genes into the nucleus of adult cells, which resulted in the transformation of these cells to something that pretty much looked like embryonic stem cells and had a similar potential to become every cell within the human body. Over the years, a number of labs have used these iPS cells then to create all these types of cells for the use in regenerative medicine. With our extensive experience of using induced pluripotent stem cells in regenerative therapies within our lab, we have found that the undifferentiated iPSCs can provoke an immune response when they're being transplanted back into the same recipient. These results were published in 2014 and made us aware of the immunogenic potential of iPSCs. Another thing to keep in mind is when you look at the surface marker expression of iPSCs, these are actually very similar to cancer cells. So combining these two properties of iPS cells, one being that they're immunogenic and the second one being that they have a surface marker expression that is very similar to cancer cells, the idea within our lab arose whether we could use iPS cells to vaccinate and prime the immune system to targeting cancer.